Hello, and today we'll be talking about cancer cells, or just cancer in general. But before we get into that, we have to start talking about things you must understand. About the phases of the cell, here we have the G2 of interphase. We have of course, the nuclear envelope here, the nucleus in purple, and this other stuff that we won't talk about. And of course, we'll have centrosomes. These help create the spindles. After the interphase, we get into what's called prophase. Here, all I want you to learn is to see that the nucleus is being basically dissolved, or packed up, you could say. The chromosomes are being combined together, or compacted. And you see the mitotic spindle forming here in prophase. Here in prometaphase, you will see the spindles stretching out on either side of the cell. This will help separate the chromosome for the two sister cells. Alright, metaphase. This is all you need to know for metaphase is the chromosomes line up on metaphase plate. And that's important because that's where you'll know which chromosome will go where for each daughter cell. Anaphase is showing the two separations. And telophase, as you see here, the nucleuses are coming back together. The cell is about to separate. You can see the cleavage furrow as I drew here. This is about to be basically cut into two, you could say. And basically, cytokinesis is the two cells actually separating from each other. Okay, let's go back to interphase for a moment. We can split this up into three phases itself. We get the G1, the S, and the G2. Now, the G1 is just for basically to know if the cell is ready to actually start synthesizing its DNA. So all the things that are required to start the synthesis of DNA, this is what G1 is. G2 is all the necessary components that go into preparing the cell for mitosis. Alright, now we talk about checkpoints. Checkpoint number 1 is at G1, and this is just basically, it could be an enzyme or some protein complex that has to be created or be destroyed in order to get past a checkpoint. And that will just help the cell not progress into synthesis of DNA. This could be like, hey, there's not enough proteins here, there's not enough amino acids to build these proteins. Checkpoint number 2, this is the end of G2 phase, and this is basically, is the DNA ready to actually be separated? Is there any errors? And this is what we'll talk about mostly. The last checkpoint before the separation of the two cells, cytokinesis, is the M checkpoint. This is right at anaphase, and we'll talk about this later on. Now, the one checkpoint that I want to talk about the most is the G2 checkpoint. We have the little purple things. This component is called cyclin, and the bottom component is called CDK, cyclin-dependent kinase. Now, when these two get together, they're called MPF, and these help regulate, depending on if they're together or separated, these help regulate if the cell should move on past checkpoint 2, or G2. And let's start off at G1, as we have a breakdown of cyclin. Nothing is being assembled. So, the cyclin has broken down completely, and CDK is the same out for most of this. Once we get to S phase, synthesis of the DNA, we have an increase of cyclin, which, cyclin comes up to the CDK, and they're like, hey, let's combine here, we can actually do something. So that complex starts to build up right after the G2 ends and the M phase begins. So during mitosis, the MPF complex will actually be the greatest concentration. And then at the end of M phase of mitosis, you'll have the cyclin start to break down again, and the complexes, the cyclin and CDK, separated from each other. And this indicates to the cell that, oh, it's time to have the cell split, and nothing is aired. So if there's an error in the system, we want to catch it at G1, but if we can't, we'll catch it at G2, and if we can't, we'll catch it at the end. Cancer comes from the errors in the synthesis process. So when there's errors in the synthesis process, the cell decides to keep going through mitosis even though it shouldn't be, and there's this error that's causing it to do so, and this is the cyclin and the CDK. The cyclin doesn't degrade, and the CDK and the cyclin stay together. So what happens is, when the synthesis of the DNA gets aired and it passes all the checkpoints, the cell will continue to divide and divide. And this is a form of the cancer. So there's two forms of cancer, and one of them is when the cells divide and divide and divide, and they keep going past these checkpoints when they shouldn't, but they have a limitation. They will stop dividing if there's not enough room for them. There's a spatial limitation, and these are called benign tumors. So they won't become tumorous, really cancerous, until the spatial factor goes away and they start dividing on top of each other. This is a malignant tumor, and this is what we call cancer, basically, because the cells have gone out of control and they will keep dividing until there's no more resources for the organ or the entire body. And good thing we have what we have here, the macrophage, because this attacks the cancer cells. Basically, you can say that it eats it up. Here we have the two cells, cancer and macrophage. What happens is the macrophage is actually read the cells on the outside by its proteins and determines that if it's a cancer one, then it will dissolve it, or it'll just break it down basically through enzymes. Another one is a natural killer cell. Basically, what this does is it sees the proteins that are marked that it indicates that this is a cancer cell, and basically inject an enzyme package that will destroy the whole entire cell, and then the macrophage will come and eat it. Or you could say that it will just recycle the material, basically, because the cell is dead at that point. Well, there are some things that people get misconception on how you get cancer. A lot of the time, cancer is a natural progression of DNA being aired in the process of synthesizing it, and this could be just caused randomly. We also have high energy electromagnetic, which is gamma rays, ultraviolet. You know, if you're going out to the sun, a sun bath, and you're sitting out there, you're introducing this high energy electromagnetic waves, and this affects the actual cells in your skin, which create DNA that is corrupt. Other types could be go to the doctor and you get an x-ray. Those are the things. Also, you get carcinogens that go into your body, like smoking. Those will cause cancer because they will interfere with the cells in a way that it will make the DNA have errors in it because of the chemicals reactions that affect the DNA. Well, thank you for watching Bioactive. You should rate and subscribe down below, and also leave a comment if you have one of improvements or suggestions, or you just have questions about other things, or you want to see something on here that you don't see, another video of something that you're very interested in. Alright, thanks again.